just want to make sure that I am, yes, I'm recording, okay, but I think that I am on the camera, not you. So
that's fine, as long as the students can hear me. That's fine.
okay, I'll lip sync as if I'm you, okay?
Okay.
All right, get going.
All right, well, good afternoon, everybody, and welcome to Microbes and Infectious Disease. I am Dr. Whitehead, although I am not actually lecturing today. Dr. Kaback is lecturing today, who I think probably most of you can see. I am your course director, your course coordinator. As you can see up on your screen, there's my contact information. The best way to contact me is to use that email address right there. That should be obviously a lowercase r. So whiteip@njms. At this point in time, I'm not putting an office up here because I'm working remotely. So I am going to be directing this. This whole course will be done remotely, but if sometime during the course of this, in the progression of this course, the state gets a little bit more organized and we get our vaccines, I will be moving back on campus and I will give you the office information at that time. So as I say, this is Microbes and Infectious Disease. This course is designed, I know most of you in this course are very interested in moving on and having careers in different healthcare careers. I'm sure some of you are thinking of medical school. Many of you, I'm sure, are thinking of dental school or PAs or PTs or OTs. And all of you, and I'm sure you will all be successful at getting into those professional schools. When you do so within your first year curriculum, you will get a course that's very similar to this, a microbes and infectious disease course. It is different than a more classical microbiology course in that this course will focus much more on the infectious disease part, all right? So this is more of an infectious disease course than it is a microbiology course. So just keeping that in mind, this is a very healthcare directed type of course. And Dr. Kaback and I and all the lecturers in this course, we all lecture in the microbes and infectious disease course, which is given to the first year medical school students here at NJMS. The content you're going to get is very similar. The degree of difficulty is very similar. The exams, which I'll talk about in a few minutes, are again very similar. So it's a really good barometer, something you can use to see how you stand relative to a first-year dental school or a medical school student. Gives you an idea how you might perform in such a course. Once you get into professional school. So we're going to be focusing on disease. We're going to be talking a lot about very contemporary infectious agents. We'll talk primarily about bacteria, some fungi, and some parasites, and some viruses as well. But the bulk of the course will be on bacteria because they represent the bulk of infectious agents. So let's go through a little bit of the administration for the course. So the course will be done remotely. So we're going to do it live lectures through these Zoom sessions. We will be meeting once a week like we're doing right now on Tuesdays. The lectures will start promptly at 2 o'clock. I booked off a 3-hour slot for this. This will be typically broken down into 2 sessions, or there'll be 2 lectures. So there'll be 1 lecture, then there'll be a break, and then there'll be a second lecture. And, some sessions will take the full 3 hours. Some will take less than 3 hours. I would count on at least 2 hours, putting aside 2 hours for these sessions. But as I say, there's many sessions that will run the full 3 hours, including the break. All of these sessions will be recorded and posted. So a folder has already been created for them. I'm sure you've all taken courses before here that have been recorded. I'm going to, so that folder's already, as I say, been made. It's available through the NJMS education portal the way you would normally access course recordings. That being said, this evening I will send out an email with the link directly to that folder, so you can all just, if you just put that aside somewhere, that would allow you to just directly link into that folder. So hopefully that will be helpful to you. There's only one session which we will not be doing live, and that is the section on January 26, Pathogenic Mechanisms and Host Defense. Dr. Beaulieu will not be doing her lectures live, all right? So her lectures have already been posted, so we will not be meeting on January the 26th. Her lectures have already been recorded, and they're already actually now present, have been placed into that folder, okay? But apart from that, all other lectures and obviously the exams will be done by Zoom. Now, the link that you're using now for this lecture, this link can be used throughout the course. This is, you know, for every time we meet. The only exception to this will be for the exams. You will get separate Zoom links sent out for the exams, because the exams will be breaking you down into smaller groups. As you can see on the slide here, there are 3 exams for the course, pretty much equally weighted. I actually set up the grade book already. I think I put it in though as 33, 33, 34. So that's the way it's calculated in the grade book. So this is just slightly off, all right? So the first exam will be worth 33%, second 33%, and the final exam will be 34%. These are not cumulative exams, okay? So, you know, the first exam will cover the information in the first section of the course, the second in the second section, and the final exam is not a comprehensive exam. It just covers the information that was not covered that was covered since the previous exam. This is the marking rubric, which we are going to start with, but I guarantee you we will not end with, all right? So this is the marking rubric that we use when we give this information, when we do this course for the medical students. That being said, using this rubric last year, I think by the end of the course, out of 70 students, there may have been only 2 or 3 students who were able to achieve a grade over A. And I guarantee you I'm not going to give out just 2 or 3 A's in this course. There'll be many more. So this will be subject to change. The course is subject to curving, but you know, I just point out, if you're in the dental school, the medical school, they don't curve. So you can, you know, refer back to this rubric at some point at the end of the course and say, you know, if you did get greater than 90%, you could say, fantastic. If I was doing this in medical school, I would have got an A for this. But as I say, I think last year, to get a grade of A in the course, it ended up being somewhere around 82 or 83% by the end of the terms. But what I'll do is at the end, after every exam, I will, you know, based on how the students performed in that exam, how you performed, I will give everybody sort of a kind of a sense. I'll give you all a sense of where your standing is in the course. Like if the course were to end at that point in time, where you would fall in terms of your letter grade. So you might find that useful. The exams will be done through ExamSoft. They will be remotely proctored. They will all be multiple choice, and they will be virtually as identical in difficulty as what you would see in a dental or medical school course. And so that's, you know, as I say, it's a nice opportunity to get a feel for what one of these a medical school course would be like. Now, in terms of exam reviews, and this is a particularly difficult issue when it comes to doing these things remotely. And at this point in time, I don't have any. Last year we were not, because we were doing this remotely and we were doing it on the fly, we were unable to do exam reviews. And by that I mean that, you know, the exam was the exam. At the end of the exam, you got your grade, and you weren't able to then go back and look at the exam itself. And now that's not particularly problematic for this course, because remember, it's not cumulative. So every exam is on new material. So it wasn't necessarily, you didn't need to see what you did poorly on the first exam to be successful on the second exam. But I feel that, I'm pretty sure there's a way within ExamSoft which we can allow you at the end of your exam to review your exam, to take a look at it before you log out. And so, but I'll give you more information about that before we get a little closer. Somebody is asking, all the questions will be multiple choice, and the answer is yes, all questions will be multiple choice. Your grades will be available and posted in EduMan, in the education management system. There's already a grade book set up in there, so you know, make sure you have access to that. If you don't have access to that, reach out to Nadine in the graduate school, and she'll make sure that she'll be able to work with you to figure out why you don't have access to that. But that's where the grades will be posted. Let's see, I just want to make sure. Canvas, all right, so the Canvas webpage has been constructed, and I published that last week. So what's up there already is there is a syllabus, the schedule for the course is posted, and Dr. Kaback's slides for today are posted. And what I'm going to do is the Friday prior to each session, I will post the slides for the next session. So that'll give everybody 3 or 4 days to pre-review the slides before they go up. Would we have a practice exam or any review session? What we were able to do, what I was able to do last year, and I'm going to do it this again a little more expandedly, I will give you some practice questions. This is probably pretty important, particularly for the first exam, because these are going to be a type of questions you haven't seen before. They're done in a clinical vignette format. So I will provide you with questions in advance of the exam, so you can just see what type of questions are going to be on there and just test your ability. Probably before each exam, I'll give you maybe a half a dozen questions, sort of representative questions, what's going to be on the exam. All right. Review session. This is a team talk course, so we're not going to bother with review sessions because we'd have to get everybody together for that. If you have questions related to the information, okay, that's going to be presented in the lectures, you should be reaching out directly to the lecturers, and you will find that they are very, very responsive. Well, will there be time allotted for question and answer sessions? Absolutely. These sessions are just like a live lecture in a way. Like today, I'm not sure how many students we have. We have 65. So since we have 65 students on for today's lectures, that's too many for people to be just, unmuting and asking questions. It'll get a little chaotic. So what I'm going to ask you today is, if you have a question, I am going to moderate the chat. All right, so I will be looking, any questions you have, just put them right into the chat, and then I will, interrupt Dr. Kaback at an appropriate moment and ask him the question. Similarly, at the end of each session, there'll be plenty of time. The lecturers will stay on as long as they need to answer any questions. And I say, if you have any questions then beyond that, when you're reviewing the material, please just reach out to the lecturers directly. And what happens is if the lecturers get a question, a clarification that they feel might be something that needs to be shared with the whole class of interest, they will send that to me and I will post it on Canvas and let you know, or I will send it out, that information out by email. Okay, that's all I have for you right now. If you think, I'm going to be here through this whole session. So if you think of any questions, you get, over the course of the next couple of hours, I'm going to be in every session. All right, even though I'm not lecturing for a while, I'll be here. And I'll generally open the sessions half an hour in advance at 1:30. So if anybody has any questions for me, they can just log on in that previous half an hour and I'll answer any questions, related to course administration and things like that. As I say, in terms of content, I'll certainly address any questions related to my content that I delivered. But the best way to get questions answered about content from particular lectures is to go straight to the lecturers themselves. And most of the lecturers in this course are giving multiple lectures, so they'll be showing up again and you'll have plenty of opportunity to interact with them directly that way. Any other questions? Okay, well, great. Well, again, thank you and welcome to the course. And I'm now going to turn the screen over to Dr. Kaback, who will be giving the introduction to the course.
Okay, I'm waiting for me to come on camera. Okay, am I on camera? Not quite.
David, you are on camera. I can see you. Yes.
You can see me. Let me get me on to onto my camera. Okay, because I don't see me quite yet, but I have to get myself in the right view. Here I am, and now I just have to pin me. So there I am, and there I am. Great. Okay. I'm sorry, I need the feedback because sometimes I gesture with my hands when I'm talking about shapes and sizes of things. So it's good to have a window of me there. Hopefully you should see my slides here on most of your screen with me as a window. So sort of make me obscure as a little mini window so you can see what I'm doing. My information, hopefully you can see my cursor is over here. And as Dr. Whitehead said, chat any questions you have and he'll wait for an appropriate time. And I'll break it a couple of times during this lecture because I cannot speak for 3 hours nonstop. So we'll have a couple of breaks and that'll be a good time where you can come on, turn on your microphones and ask me a question. And again, you can also email me any questions you have at kaback@rutgers.edu. And in most cases, I will end up putting those questions onto the Canvas website for everybody to see, unless they're private of some kind. And so unless you object to me posting it anonymously, let me know. And otherwise, if you ask me a question on a chat or an email, I'll try and get that onto the post. So welcome to Microbes and Infectious Diseases. I'm going to tell you about a lot of sickness. And let's start off with the fact that pneumonia and diarrhea are incredibly horrible causes of fatalities, especially among children, especially in underdeveloped countries, but also what you're going to be hearing about are diseases that affect plenty of people here. And of course, we're now in the worst pandemic of anybody, pretty much anybody who's alive, who's less than 102 years old, can remember with respect to this COVID SARS-CoV-2 epidemic. And anyway, the bottom line is infectious disease is a horrible shortener of life. And you can see these WHO statistics, and we'll be soon back in. Actually, we never left. We're not haven't left the WHO yet, and we'll soon be back. And as you can see from these numbers, the number of deaths worldwide, in fact, infectious disease accounts for 1/4 of all fatalities. And these are, when I should say infectious, I'm talking about microbial diseases, stuff that we will be hearing about during these class sessions. And you can see, by the way, that the statistics for respiratory infections used to be only about 4 million deaths per year worldwide. It's now, due to a single virus, so far up to more than 6 million are respiratory infections, since COVID mostly affects the lungs, but COVID also has some other effects. It's a much more infectious disease that affects more than the lungs. And here are the others: HIV, diarrhea, tuberculosis. Fortunately, venereal disease like syphilis a little bit goes down, used to be much more. But you can see million fatalities due to malaria, 1.6 million due to tuberculosis, 2 million due to diarrhea, believe it or not, which can be a fatal disease. You lose all your water and you die of dehydration. All right, that said, this course we're going to be talking about bacteria, eukaryotic pathogens, including fungi and parasites, and viruses, which are debatable as to whether we want to call them alive or not, since they require a living organism to replicate in. So beginning with bacteria, okay, we can see a typical bacteria next to a red blood cell, and they're anywhere in size from about 1/2 micron to 2 microns. Here, this one here is about 1.5 microns shown here next to a red cell, okay? And you can see that many bacteria can fit in a single red cell, and red cells are one of our smallest cells. And here it is enlarged over here. And that bacteria, of course, it makes viruses seem even smaller. You can see the relative size of a virus to a bacteria as shown here. So we're going to be talking first about bacteria, which, as you probably already know, basically are prokaryotes, which means that they don't have a nucleus, okay? There's no nuclear membrane, no nucleus. The DNA is more or less naked within the cytoplasm, not quite, okay? And they're typically single-celled, and again, very small. We'll also be talking about fungi, and when we talk about fungi, we will be talking about our first eukaryotic pathogens. Actually, I think we have protozoa before fungi. I have to look at the schedule, but these are eukaryotes, meaning they have a nucleus. Okay, you can see the nucleus right over here in a yeast cell. And what we're talking about when we talk about fungi are yeast and molds. And mushrooms actually are a form of mold, a very highly organized mold, as opposed to what you used to seeing growing on that food that you left in the refrigerator for that 6-month-old jar of mustard or mayonnaise or yogurt that you left in the refrigerator and forgot about. And you'll see one of these guys growing on it. And in the microscope, it looks like that. And we'll be hearing a lot more about that. And these things can also cause, and certain strains of these can also cause infections. Usually, very often, they're superficial. But when they're not superficial, very often they're what we call opportunistic, meaning only people who are immune compromised get infected by it. But then there are also completely pathogenic fungi that one can get an infection. The next thing we'll be talking about are eukaryotic parasites, meaning protozoa, like this guy here, this is Naegleria, which basically if you go swimming and in an infected pool and it swims up your ear, it somehow can get into your sinuses and invade your brain, causing a fatal infection, Naegleria, or another parasitic infection is malaria. Okay, oh, that's poetic, Naegleria, malaria. I didn't realize that. So we can say, and in addition to protozoa, we'll also be talking in terms of eukaryotic pathogen parasites, worms, okay? And these can invade your digestive system or your muscles. And these worms are covered in a microcourse because they usually have a microscopic egg cell stage or a very small larval stage that is microscopic and can infect you. So these are eukaryotic parasites, and you'll be hearing a lot more about that. And finally, we'll be talking about viruses. And for those of you who don't know, virus comes from the Greek word for poison, because people used to think when you got a virus, it was just like somebody giving you hemlock down your ear, like Plato or Socrates, whatever, etc. You feel rather sick as if you're poisoned. And of course, these can't replicate without a host cell to replicate, and they cannibalize the host cell replicative machinery like ribosomes, et cetera, so that they can make their own protein and often encode their own replicases so that they can make their own nucleic acid. And these were originally defined, historically, a virus was defined as a infectious agent that would cause disease, that basically when you put the liquid from a diseased person through a diatomaceous earth filter, it would come out and you couldn't see it because you couldn't see anything in a microscope, because at this point they didn't have electron microscopes. They had only optical microscopes, and they were completely invisible to an optical microscope. So these were infectious agents that were essentially found in the filtrate after filtering them, whereas bacteria in the early age of microbe hunters were stopped by these diatomaceous earth filters, and you could actually see them in the microscope early on. And now we know that there are some bacteria that actually can still pass through those filters, like mycoplasma, and you'll hear about mycoplasma pneumonia from me, and some urinary tract infections that also are these very small bacteria called mycoplasma later on. But basically, viruses are much smaller than bacteria, which are in general about 1 micron long, we say 0.5 to 2 or 3 microns in size. A virus basically has nucleic acid and protein, and there are a class of viruses that don't have envelopes, like adenoviruses, which are one form of cold virus. Okay, non-envelope virus like polio, Coxsackie, as I said, adenovirus, are basically a protein coat with the nucleic acid inside them. It could be DNA, it could be RNA. Okay, and these actually are more stable to the air than the other class of viruses, which are what we call envelope viruses, like flu, and of course, coronavirus, which you all know has a lipid envelope with a lot of spoke proteins, looked like that, shown here. Okay, envelope. And these are much less stable to the air and to dry than these simple viruses that don't have a envelope, that don't cannibalize the cell's envelope when they are blebbing out new infectious particles. Okay, so now I'm a firm believer in art and science, and what I'd like to do is just read you a little bit of poetry, and this is a cute little story that's in your notes about how I once met the protege of W.H. Auden, the very famous poet, and told him all about viruses and actually viroids. And 2 days later, he mailed me this poem that was ultimately published and I'll just read the beginning of it. A virus like a finger is a bone with protein coat, a dissonant dead ringer, a cellular sour note that sometimes will linger in eye, ear, nose, and throat, depositing its stinger to get genetic goat. A class called retrovirus is culpable in tumors. Like gossips, they hotwire us with metastatic rumors. Some rapidly retire us, while others are late bloomers that patiently admire us in neoplastic humors. A viroid, on the other hand, and I'll stop there, and you can read the little story in the notes, which brings me to the fact that I like to add a couple of notes to the slides that are initially posted on the Canvas website. Okay, these usually contain additional information that I may or may not cover. It's usually not essential information. Sometimes I put the information on the same slide. So if you're following along on the Canvas website, you may see something a little bit different, and that'll contain my notes, but it's mostly interesting stuff that is not necessarily required. The other thing is I like to add these stars to a bunch of slides that you'll see. And these slides I consider the most important in the presentation. So when you're studying, try and concentrate on the material that's in there. I'm not telling you to memorize them, but they contain important information that you should make sure you're familiar with, okay? One of the things that always comes up in this course is how much do I need to memorize, and do I need to memorize every drug for every organism? And the answer is, in most cases, that will be described by the lecturer. For my lectures, the answer is what you need to do is remember sort of some general rules with respect to drugs. And when I tell you a drug isn't going to be good, you can assume that virtually all the other antibiotics do work, stuff like that. So again, it's lecturer dependent, and they'll be specific, and you can certainly ask me that question again, depending on how you feel. Okay, so that said, let's begin with infectivity of organisms. And one of the things that are really important that is the fact that we are not alone in respect to our human cells in our bodies. Basically, there are about 30 trillion, 3 times 10 to the 13th human cells in a human body. And there are an equal number of bacteria and a much smaller number of fungal cells living on our bodies that are very much part of our digestive systems, part of our skin flora. This is normal flora. They're virtually everywhere in your body, certainly underneath your arms, in your nares, right over here, you have a bunch. In the creaks of your eye, wherever you have a fold in your skin, you're going to have an anaerobic environment that may produce a different class of skin organisms than in the parts of your skin that are completely exposed and aerobic. And all you have to do is take a petri dish, scrape it with virtually any part of your body, and I can pretty much guarantee, unless you've had a shower and washed every part, you would essentially come up with bacteria growing and perhaps a couple of fungi growing on that petri dish. Okay, and this happens as soon as you're born. Basically, you come through that birth canal and you come through the vagina, or even the vagina is an open area that has lots of normal flora in it. And even if you're come by C-section, babies come by C-section, as soon as the first person who picks you up without wearing gloves, like your aunt or your dad, etc., you're going to get infected with, start getting to accumulate your normal flora. Okay, virtually everything you eat is contaminated. Okay, hopefully not with much and not with pathogens. We'll hear about plenty of pathogens that are found on contaminated food in this course, but basically bacteria are all over the place. Anytime you inhale, you are probably inhaling a fungal spore, likely, and certainly some airborne bacteria. Remember that whatever you're inhaling is something that likely was exhaled by somebody else, at least part of it. And hopefully any particulate matter and any droplets have sunk to the ground by the time you've inhaled it. On the other hand, as we know, that isn't always the case. And we have to be careful and wear our masks when we're in public now. So basically, normal flora is virtually everywhere. And for many of these bacteria, there are establishment into specific niches in the body, places where they can be, including the vagina, the lungs. Your urinary tract should be sterile, for example. On the other hand, your ears are quite exposed, as is your nose and your oral pharynx, the upper part of your respiratory system. Once you start breathing things in, you have mechanisms that push things out. You have a whole set of cilia that are trying to push foreign materials out. But unfortunately, when normal flora gets into the wrong place, or we essentially inhale or eat a pathogen, or it gets in our skin or in a wound, we end up with abnormal flora. And I say fauna too, because we're dealing with parasites, protozoa, as well as viruses. Again, you can eat them, you can inhale them, you can get it in a wound. And as I told, the good news is that many microbes, including viruses, are not very resistant to drying. We'll be hearing about bacteria that are so resistant to drying that you can virtually autoclave them, and they will still, a small percentage of them will still remain alive, even if you put them in a boiling water, if they maintain that survival thing. So you'll hearing about all sorts of ecological niches of these bacteria and viruses. And again, one of the other thing is, how many bacteria, how many viruses do I have to inhale, et cetera, to get sick? And the answer is it's highly variable, it's highly disease specific, and it's highly patient specific, depending on somebody's immune state of somebody's immune system. But for example, if you have the flu and you're shedding virus, 1 milliliter, okay, a fifth of an ounce, sorry, a 20th of it. Let's see, a milliliter is about, I think about a 30th of an ounce, if I recall. I have to always forget that. So about a milliliter of respiratory secretion, if you're having a cold, that's not much when that's one nose blow. That can have anywhere from 10 to the 5th to 10 to the 6th, a million viral genomes in them. Now, a good news about these respiratory secretions, by the way, is that for these respiratory viruses like coronavirus even, not all the particles are actually infectious. So again, it depends on the virus. And there are some viruses that 1 to 3 infectious viral particles is all it takes to get you sick. May have a lag by the time it makes enough viruses to give you an immune response to make you sick. But there are some viruses that are highly, highly infectious. And of course, we've all just heard about this more infectious coronavirus, which apparently binds to the receptor a little bit better than the old strain of coronavirus. So again, one of the important things when we're talking about contagion and infectivity, and I want to remind you, when you can see these beautiful images, dark field images of what a sneeze looks like, how important it is to wear your mask. You don't want to have a 10 microliter or even a 1 or a 10th of a microliter secretion here, because one of these femtoliter particles can still contain many viruses. Okay, and again, if one of them is infectious, it could cause. And again, air currents from a warm body in a hospital bed can keep the smallest droplets airborne for much longer than they would do in maybe in the outdoors, and certainly won't be depleted by the very volume that they're in. So being inside is a place where contagion abounds much more than being outside, where the entire atmosphere will dilute whatever's there and hopefully dry it out.
Dr. Kaback, there is a question. Why are the non-enveloped viruses more stable in the air than the enveloped ones?
That's a very good question. And probably because they do not depend on, I'm just hypothesizing that they do not depend on water in their structure as much as the envelope viruses. And in other words, once they become dehydrated, they're already, and this is a big guess, is that they're already dehydrated, whereas envelope viruses are not dehydrated. Their envelope viruses vary, not entirely, but many of them have RNA genomes that probably once they lose their water, probably are immediately attacked by oxygen and other things that render their genomes mute or non-infectious. But that's, maybe you can add to that, Ian?
Yeah, so yeah, and we'll certainly we'll be talking about this a lot more towards the end of the course when I'll be giving the lectures on the viruses. Yeah, you just have to, think about a lipid-based cover versus a protein-based cover. So your caps, if you're a enveloped virus, you're lipid-based, and they're just so. A lipid is so much more sensitive to things like pH and dehydration and all kinds of different environmental insults versus a protein core, which is going to, a naked virus, which doesn't have the envelope. It just has this this protein shell on the outside. These can be, made up of proteins that are very difficult to denature. And so it's just a much more durable cover. And so, if you're talking, for example, about infections of the GI tract, where you have this super low pH, there's no way an envelope virus could survive in that kind of environment, whereas something that has a hard protein shell over its surface is much more likely to stay intact and to stay infectious. So that's really the difference. Somebody just said, does the envelope play a role in reducing drying? I wouldn't say so much, but in fact, I think Dr. Kaback is correct that the envelope would actually probably make you much more sensitive to desiccation.
Yeah, no, an envelope, basically when you dry the envelope, the lipids would essentially invert and probably lose their structure immediately and essentially render the genome without its they would lose the genome would essentially come out of it and would go in the capsid. And basically the viral infectivity is dependent upon that intact, in most cases, that intact membrane going and hitting the receptors of the cells that it's going. So you need to maintain that intact membrane that wouldn't be intact if you dry down. If you think about a lipid droplet outside like this, it would lose its, if it dried down and dissolve, the membrane would no longer be integral, and it would release the viral nucleic acid and capsid where it wouldn't have receptors and couldn't get into the cells. So that's just speculation. All right, any other questions, Ian?
No. I think we're good, David.
Okie doke. All right. So with that introduction to general infectious diseases, we're going to switch over and begin talking about bacteria. and that's what the rest of the lecture is going to be about. We're going to introduce bacteria, not so much talk about specific organisms, but teach you a little bit about bacterial physiology, their cell biology, and their genomes. And here are the content objectives, which you can read on your own. It's a good time to read these objectives after you've studied and make sure that you've understood everything. Okay, so beginning with bacteria, what we are talking about bacteria is bacteria are organisms, and they need to be classified and described so we can differentiate one organism from the other. And knowing that you have one bacteria, if you find one bacteria in a certain spot in somebody who's presenting with a certain syndromes, you need to basically know that that bacteria is a possible cause or a probable cause of whatever disease they are getting, and not due to normal flora. So you have to be able to describe that bacteria, classifications of it, and then you can say, aha, that's the cause of it. And if I treat that bacteria with the appropriate antibiotic that I know that bacteria is sensitive to, then I have a good shot at curing the infection. Or alternatively, if I blow that call and I use the wrong antibiotic, or one that the bacteria is not sensitive to, then I've screwed up, and the patient gets sicker and perhaps dies. So we need to classify bacteria. And so, for example, when we're talking about classification, every bacteria has a first name, which is the group of bacteria or the genus, and then a species name to it. So here are two species that belong to the genus Neisseria, Neisseria meningitidis, which of course causes meningitis, but also causes a lot of other kinds of infections, and Neisseria gonorrhea, which causes a urinary tract infection. It's a sexually transmitted disease that you've all heard of, commonly goes by the name the clap. Here is a non-pathogenic species of Neisseria called subflava. And I'll tell you why these things are called Neisseria at all. But we actually now have, for every genus and species, many bacteria have a number of subspecies. Sometimes these are called strains. Sometimes they're called serotypes if the identification is based on using a specific antibody that only reacts with one versus another kind of species, usually to the outer surface of that bacteria. So for example, not all strains of Neisseria meningitidis cause meningitis, only 5 of which cause disease. We speak about E. coli strain 0157, subspecies H7, which is a highly pathogenic strain of E. coli. Everybody has E. coli in their digestive system, in their colon, but only having this guy is going to cause you a severe bloody diarrhea that you might die from. So you don't want this strain in your hamburger meat. And you can imagine, you can find E. coli in virtually any swimming pool, in any ocean, in any freshwater pond, but you certainly don't want to find E. coli 0157 there. So these are subspecies. Okay, so when we are describing bacteria, and we want to say something about Neisseria or any other kind of bacteria, the second thing that we talk about, okay, and I'll tell you the first thing in a minute, but the second thing we describe about it is its morphology, its cell morphology, because that's the first thing we observe in a microscope. And we describe that morphology as either cocci, okay, which comes from the Greek word for berry. And sometimes these cocci form pairs, in which case we refer to them as diplococci, double cocci. If they are in chains, we refer to them as streptococci, which are twisted comes from the Greek word for twisted, because you see these twisted coils of multiple cellular bacteria. And sometimes they occur in clusters, and this comes from the Greek word for a bunch of grapes called staphylococci. Okay, so chains and clusters or pairs or all kinds of cocci. We also have rod-shaped bacteria called bacilli, comes from the Greek word bactron, which means a staff or a rod. And to confuse the issue, we sometimes have very short bacilli, and those are referred to as coccobacilli. Okay, so bacilli and coccobacilli. And then we have spirochetes. These are spiral bacteria. They're spiral because they have a flagella, a little thing wrapped around or the sort of the inside of their space that cause them to have this spiral form. And we'll hearing a lot about spirochetes that cause things like venereal disease, syphilis, as well as cool things like leptospirosis or Lyme disease that I will be talking about in a later lecture. And finally, we have Vibrios, which some people call bacilli because they're sort of rod-shaped, but they're really comma-like. And they're called Vibrios because they vibrate. And the reason they vibrate is they have flagella that move them back and forth, causing them to vibrate. And Vibrios, you've all probably heard of Vibrio cholera, as well as a number of Vibrio species that inhabit shellfish. So if you like raw oysters like I do, pray that they are not contaminated with a Vibrio that's going to cause diarrhea. So that's the second thing when you're describing a bacteria that you learn. What's the first thing? Well, generally, before you say it's a cocci or a bacillus, you generally say whether it's a gram-negative or a gram-positive bacteria. So things will be gram-negative bacilli or cocci or gram-positive. And this has to do with how they react to the gram stain, which is a stain that consists of crystal violet or gentian violet, which is actually an old-fashioned drug for getting rid of bacterial infections. And it stains bacteria blue, okay? The difference is when you're doing a gram stain, this was invented by a guy named Gram. His name was actually Hans Christian Gram, not to be confused with Hans Christian Andersen, who invented The Little Mermaid and great songs like Under the Sea. Basically Hans Christian Gram invented the gram staining procedure where one class of bacteria will take up the gram stain, but as part of the gram staining protocol, you actually wash out the stain. And a lot of bacteria will learn that stain washes out, those are gram negative. And then you use a second stain called safranin that stains those bacteria pink. So when you look at a gram stain species, bacterial stain, you'll see gram-positive bacteria will stain blue, and gram-negative bacteria will stain pink because they only pick up the safranin stain. I'll tell you why that is. Then there are a bunch of bacteria that are refractory to gram stain, don't stain at all, and those are like mycobacteria. And mycobacteria you can see because they stain with carbylfuchsin, which is called the acid-fast stain. Okay, and then there are bacteria like mycoplasma that are so small you really can't see them. They're barely dots in a oil immersion microscope, so you really can't tell what they are. And you'll see they won't stain by the gram staining mechanism as all, as you'll see why when I talk about mycoplasma later. And then there are treponema, which are really gram negative, but they stain pretty poorly with the gram stain at all. So they're very hard to see even with safranin, but they still really are classified as gram negative if you're asked. Okay, so we have gram positive cocci, gram positive bacilli, gram negative cocci, gram negative bacilli. And here are probably the most common genuses of all of these gram-positive, gram-negative bacteria that you're going to hear about. Okay, so make sure you remember that these species, these are genera, these are the genuses, that you remember these things. Is a good thing to memorize. Okay, so in addition, so when we talk about bacteria, the first thing you'll hear is, well, basically we took a sample from somebody's lungs and did a gram stain, and we saw gram-negative bacilli in there. Okay, so that tells you immediately, if you're a microbiologist, that gram-negative bacillus from a lung infection, that's a good possibility that they might have something like Haemophilus. Okay, on the other hand, if you heard gram-positive cocci, diplococci, you'd say, that person likely has pneumococcal, and so in 10 minutes, somebody has a vague idea about what bacteria is causing them just from the gram stain and the morphology of it. Okay, nevertheless, and in the old days, interestingly enough, medical students were who would do the bulk of the gram staining. So when you got a sample in the clinic from somebody who's sick, the first thing you would do if you were in a teaching hospital is you would send the medical student out to do a gram stain. So you better hope that they knew how to do gram stains and could do them accurately, because hearing that you had a gram-positive diplococci in there is going to be really important for the treatment of pneumonia, because pneumonia, as long as the patient isn't allergic to penicillin, it's somebody allergic, gram-positive diplococci, likely they have pneumococcus, and you're going to treat them very aggressively with penicillin because it works a whole lot better and faster than Zithromax on pneumococcus. Okay, so this was a very useful bit of information, and you want to basically put somebody on antibiotics as soon as possible, because they work really quite fast for many diseases. Same thing like strep throat. Okay, somebody gets does a gram stain on a throat and sees gram-positive streptococci, it's likely they have a strep infection. And if they are not allergic to penicillin, basically if it's a kid and you do a strep test, then there are faster ways to do this. They have these quick strep tests. But if you found strep and you put somebody on it, and it's the afternoon, the next day that kid is back in school because the strep is gone. So being able to identify something quickly by a gram stain is still a very effective thing to do. Unfortunately, medical students have lost their license to do it. They never had one. And now for legal reasons, they essentially have to send all samples down to a lab to be done by a professional lab person. So the days of medical students being the primary doer of gram stains, those are no longer, and we no longer demand that of our medical students, but we did for many, many years. All right, so in addition to gram-positive and gram-negative, we have descriptions of the shape of a bacteria, which I told you about, okay? And the gram stain and the shape, gram-positive, gram-negative cocci bacillus. And then we'll describe it by various things about how that bacteria is grown. We'll start to culture that bacteria. We'll plate it out on a blood agar plate or a chocolate agar plate or a McConkey's plate, and we'll learn whether it requires or doesn't require and is killed by the atmosphere, if it's an obligate anaerobe or an obligate aerobe. And then we'll also learn whether some bacteria you'll hear are facultative anaerobes, means they can grow both with and without atmospheric oxygen. Okay, and we'll hear about the biochemistry and genetic classifications, whether they, for example, have certain enzymes. We'll hear about things like cytochrome oxidase as being an organism is oxidase positive or negative. So all of these are important. And then finally, we will determine subspecies with serology. We'll use antibodies to specific types, subtypes of bacteria to see whether we have a pathogenic strain of that particular organism. And then finally, other things that can happen is that some organisms are very resistant to drying, like anthrax and certain anaerobes produce spores or endospores. And you'll hear about aerobic and anaerobic spore-forming organisms. For example, Bacillus cereus is a common food contaminant, and it's an aerobic, gram-positive, motile spore-forming bacillus, okay? In contrast, E. coli is a gram-negative lactose fermenting, see biochemistry, lactose fermenting, it makes beta-galactosidase, facultative anaerobe. So these are all ways that we describe bacteria. And here is a list of a large number of other characteristics that are sort of submorphology categorizations of what a good microbiologist will learn. This is not, notice the slide does not have a star. It's not something that you know, you need to know. But I have highlighted in here most of the organisms that you will have to become familiar with, not all, but most of the organisms that you'll have to become familiar with over the next, during this semester. Okay, again, you'll notice nothing here, no slide. This is not a slide to memorize, but when you're studying, make sure you have heard of all of the organisms shown on this slide. It will help in your studying. Okay, with that said, it is now 3:02, and I think a good time to stop. If you have any questions, I'll take them. Otherwise, so let's begin in about 7 minutes, and we'll continue on. All right, so take a break, and I'm going to go out and take a break. If there are questions, I'll stick around for another. Dr. Whitehead, do we have any questions?
I'm not seeing anything right now. So yeah, so let's take a break. We'll pause our recordings and we'll start up again at 3:10.
All right, please remind me to
restart. I will.
Yeah, let's see. Pause. Resume recording. All right, we are back recording. Excellent. All right. I hope everybody's back. I haven't looked at the participant list to see. We still have 65 people who are logged in, which is very nice. They may be recording this. We don't have them on their screen. I always wondered how many of them are doing Ferris Bueller's, where they put a picture of them working assiduously on the screen when they're in fact out playing tennis. But that said, let's begin. And going back to our talking about bacteria. And again, I want to tell you, emphasize how important it is to identify the source of infection and able to differentiate between a pathogenic organism and normal flora when you're diagnosing a bacterial infection. And tell you a little bit about how we identify bacteria in a clinical lab. In the old days, and still done today, not completely eclipsed by more modern methods, is microscopic observation of clinical samples, okay? Finding and identifying a gram-positive or gram-negative bacteria, and then having a presumptive diagnosis based on its gram stain and its shape. A good microbiologist is pretty good on it. And then essentially doing culturing that bacteria. And if it's culturable, okay, doing a number of metabolic, biochemical, serology tests on it, looking for specific antigens or enzymes by biochemistry in order to more appropriately classify that bacteria as being a specific pathogenic species. And again, identifying that subtype via serology using antibodies to specific pathogenic strains of that organism. This is what I refer to as classical diagnosis. Okay, finding that. And as opposed to contemporary diagnosis, which still begins with the microscopic observation of a clinical sample. Again, that takes 10 minutes to do and it doesn't cost very much money. Okay, and now what we do is we analyze our samples via PCR. Okay, these are multiplex PCR, so we're using primers from 95 up to 95 different organisms that each produce a specific size or very often color, if we use a colorimetric test, that is going to, if that organism's genome is there, going to provide a PCR signal that's positive. We're now even doing mass high throughput DNA sequencing for bacterial identification. That's coming out, not done routinely yet in most places, but as we get better and better and the machines get better and better, DNA sequencing will become more the norm. But certainly PCR is the norm for bacterial identification. Okay. Another thing is that if an organism is culture positive, if you take a sample and it's culture positive, okay, one of the fastest way now of identifying that culture positive organism is by the mass spec. This is mass action, time of flight, mass spec reaction, and every organism, due to specific molecules that it's making that have a specific molecular weight, will give a spectrum and give a fingerprint that is typical of that organism. And these are more and more coming online.
David, sorry to interrupt. Can I pause you just a second? A student is saying that they cannot hear you, but is anybody else having that issue? Okay, so other people are saying everything's fine. So Yoku, it's got to be something going on from your end, okay? Because the audio, I'm hearing him fine. Everybody else is hearing him fine. Sorry,
Okay.
David, go ahead.
No problem. All right. Are we back on recording or you never stopped it? Yes, I see.
Never stopped recording. You're good.
We're recording. Okay, we are recording. Thank you. All right. I apologize. I hope that student is able to get the recording to listen to me. Anyway, let me just go back to the fact that we were using mass spec as a means of every organism has a specific set of fingerprints that it does when you just take a little bit of that culture and you put it on a mass spec. It gives you a complex fingerprint that is specific for an organism, and this is coming online. The only problem with mass spec, and for that matter, all of these things, is that less than half of all bacterial infections actually produce a culture that is meaningful on a petri dish. As I mentioned, virtually everything you sample will grow something, but whether you're growing the pathogen that and is only about half of the bacterial infections actually produce a culture positive. And that's because by the time you mostly show up and are being sampled, your immune system is already kicked in with all these super antigens, and it started swallowing up that bacteria. So that organism may already be present within cells but though it's not going to be culturable. If it gets swallowed by a macrophage and it's an obligate intracellular organism or facultative intracellular organism, it may be growing within your macrophages and capable of producing a lethal infection. So again, not all bacterial infections are culture positive. And
David, there's a question related to that.
okay.
Can you repeat? Does culture positive refer to growth on a standard agar, or does this refer to all different growth mediums?
Pretty much all different. I should say, basically, when a sample is gotten in the clinic, okay, it is routinely cultured on blood agar, chocolate agar, and a McConkey plate, which basically selects for gram-negative bacteria. When you culture it on those three kinds of media, I would say about half the samples, maybe a little bit less than that, will produce bacterial growth of the organism that is causing the clinical syndrome. That's the best. I hope that that answers the question. Okay. So that's the routine identification. No, the answer is that routinely you don't, there's special agar, for example, for Legionella, okay? That unless you suspect Legionella, that organism, you don't necessarily plate on that. But if you got a negative culture and you suspect Legionella based on clinical symptoms, you're going to essentially the next day put it on Legionella. But in fact, you'll have seen it at that point. Multiplex PCR is really what's going to come in. And that's where PCR testing is very important now, because you're testing for 95 different strains of bacteria, different kinds of bacteria, and you're going to pick it up on PCR, even if it's not going to grow out on an agar plate. Okay. And if somebody comes in with pneumonia and they're immunocompromised and there's no evidence of anything growing, and then you see on PCR, you find out that you have Legionella in the Legionella DNA in the PCR thing, it's very likely that patient has legionellosis. Okay, so that should answer that. Before we had PCR, it was the art of medicine that enabled you to diagnose this more than anything. I hope that answers the question. All right, let's start with talking about bacterial structure. Okay, here's our favorite generic bacteria, showing all the different possible parts of a bacteria. And let's start by talking about the intracytoplasmic components. Basically, a bacteria has in its cytoplasm about 1,000 different enzymes. There are some bacteria that make as few as 500 proteins, but in general, most bacteria have about 3,000 genes on their genomes that encode protein, and about 1,000 of these are enzymes. Another 2,000 are other proteins that don't necessarily have catalytic functions, but they have some structural or auxiliary or cofactor reaction with respect to bacterial metabolism and bacterial physiology, okay, structural, et cetera. Okay, in addition, we find nucleoids. These are the double-stranded, usually circular DNA molecule, shown here, all condensed up, highly condensed. We call that a nucleoid. And in bacteria, it is not encased in a nuclear membrane. There's no nucleus. It's free in the cytoplasm. And if we were to lyse a bacteria on an electron microscope grid, we will see that bacterial genome come out like this in all of these, what we refer to as supercoiled loop domains, shown here. Okay, so here's our bacterial genome. This is the E. coli genome. It's about 1,400 microns. That's 1.4 millimeters of DNA compact jammed into that nucleoid. In addition, In addition, we have very many bacteria have these little autonomously replicating circular DNA molecules, mostly circular, that we refer to them as plasmids. Okay, very often they contain genes for antibiotic resistance. Sometimes they encode toxins. Some of these plasmids are very important in the ability of a bacteria to transfer genetic information, which we call sex, okay? And about 15,000 ribosomes that are going to be involved in making proteins. And ribosomes are, of course, a very important part of antibacterial antibiotics, because a large number of antibiotics go for inhibiting protein synthesis by interacting with one or the other subunit of that ribosome. Okay, when a bacteria divides, okay, so here we have following cell growth, the cell will elongate or get bigger, and the DNA will replicate, shown here. And ultimately, the two chromosomes will separate. We have a very elaborate partitioning system that enables the two chromosomes to move away from each other as the membrane elongates. It doesn't use a mitotic apparatus, but in fact attaches itself to the membrane indirectly. And as the cell grows out, the two chromosomes separate. It involves a lot of topoisomerases and other enzymes. And finally, we get septum formation, the formation of cell membrane and cell wall that divide the bacteria into two distinct cells that sometimes will stick together for a while, but ultimately can produce a single bacteria capable of producing an entire colony. And bacteria, when they grow, we say, and when they provided with a lot of the appropriate growth media, will grow exponentially, meaning one cell will give rise to two cells. Each of those cells will give rise to two cells. And now two cells will give rise to four will 8, 16, et cetera, which produces a very rapid growth, as we can see here.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in two. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
All right. So 5,000 billion is 10 to the 23rd, or a mole of bacteria. Okay, I didn't check that calculation, but it's pretty, the replicative potential of microorganisms is pretty limitless. And fortunately, they have to compete in the environment with everything else. So they don't completely take over the biosphere, but they are very much an integral part of it. All right, let's go back to talking about cell structure. Okay, and the first thing we want to hear about in cell structure, as we've spoken about what's inside the cytoplasm, but now we're going to talk the outside, is the cytoplasmic membrane, and next, the cell wall. So here are gram-positive and gram-negative bacteria. And I'll tell you the differences, because that is mostly due to the cell wall of that bacteria as to how they will stain. Okay, but let's first look at the cytoplasmic membrane. Okay, bacteria, like all other living organisms, contain a typical plasma membrane with a lipid bilayer shown here. Here we have our lipid bilayer with aqueous phases on here, shown here, and shown here. And there are plenty of proteins that are involved in transport of solutes and other functions shown here on either side of it. All right. And so typical lipid bilayer. The only difference is that whereas in our lipid bilayers, we have cholesterol or sterols, okay, bacteria, with one exception of which are mycoplasma, okay, which I'll tell you about at a later lecture, bacteria don't contain sterols in their cell membranes, which means that their cell membranes are much less or much more sensitive to osmotic stress than eukaryotic higher cell membranes, because if you put a higher cell membrane, if you put a higher cell into a slightly hypotonic, lower osmotic pressure solution, that membrane will take up water, but it will not expand that much that the cell will break open, because the membrane has integrity given to it by that cholesterol. In contrast, when you have a sterile-free membrane, like in a bacteria, okay, the water will rush in, cause the cell to swell, and ultimately the cell will burst, okay? But fortunately, bacteria, okay, bacteria which have to live under large variations of osmotic pressure, whether they're in fresh water or in your body where the osmotic pressure is high. In fresh water, the osmotic pressure is low. Fortunately, bacteria are surrounded by a cell wall, okay? And the structure of that, simplified, is shown here. And that cell wall is made out of what we refer to as peptidoglycan. It is a polysaccharide cell wall. And here is a gram-positive cell wall, and here is a gram-negative cell wall. And the gram-negative cell wall is actually much thinner than the gram-positive cell wall. But the gram-negative cell wall actually has an outer membrane that we'll talk about shortly, okay? But let's first talk about the actual cell wall, which is comprised of what we refer to as peptidoglycan. Okay, peptidoglycan means we've got a peptido portion, which is a small oligomer of peptide, a small peptide oligomer of amino acids. And you'll notice that this peptide amino acid actually has D amino acids in it. It's usually a combination of D as well as L amino acids, whereas in all our proteins, we have only L amino acids. The bacterial cell wall actually in this peptido portion has some D amino acids, okay? But it's called peptidoglycan because it also has these peptides are actually bound to a polysaccharide, otherwise known as a glycan, because it's the glucose analog, okay? And it is a copolymer of N-acetylglucosamine, shown here. So you see this N-acetyl molecule over here. Here we have a nitrogen and an acetyl group, N-acetylglucosamine. This is glucose here. And the other subunit of this copolymer is known as N-acetylmuramic acid, because it also has this N-acetyl group on this amino portion of this protein, but it also has this ether group over here that attaches it to this short peptido part. Okay, now this is a structure you don't have to memorize. Okay, notice no slide here. You don't have to memorize the structure of a cell wall and whether it's an alpha or a beta linkage of the polysaccharide. What you should know is this part here. Okay, we have this copolymer of N-acetylglucosamine and N-acetylmuramic acid with peptides, D amino acid containing peptides. And furthermore, when we have two layers or adjacent layers of polysaccharide, they are cross-linked via these two peptides, by the peptidoglycan. So the cell wall, if you look at me here, is cross-linked together to maintain its integrity. So we have these threads that are cross-linked together to form this solid matrix, okay, of peptidoglycan to form that cell wall. Okay, now this is going to be, as we'll see in a minute, the source of why penicillin kills bacteria. Okay, so parallel strands that are cross-linked. All right, so let's first talk about the gram-positive cell envelope. Okay, notice the starred slide here. Here we have the lipid bilayer over here. And what we find on gram-positive organisms is about 40 layers thick of peptidoglycan, okay? Very thick polysaccharide, which actually captures the gentian violet, that purple stain, and makes these things blue because it captures a lot of this gentian violet to make it blue. And so after you add gram stain to it, after you add gentian violet to it, and you wash the slide with alcohol, which is what you do, the stain doesn't wash out of this peptidoglycan because it is so thick. In addition to this polysaccharide, we see here teichoic acids, wall teichoic acids and lipid teichoic acids, that's shown here. Here we are. And we have these lipoteichoic acids that extend from the lipid bilayer. These are also polysaccharides, okay, sugar polymers, okay, that extend through the peptidoglycan and decorate the outer surface of the membrane with a, if you think about it, sugar is soluble in water, so it's a hydrophilic layer over here. And we have some teichoic acids, and again, they're sugar phosphate, I should say. There are some teichoic acids that just come from the peptidoglycan, as well as other teichoic acids that extend from the cell membrane. These are referred to as the lipoteichoic acids because they're covalently bound to the lipid. Now, it turns out that these teichoic acids are considered virulence factors. Very often they're involved in adhesion to mammalian surfaces to make these bacteria a little bit more infectious when they adhere to certain tissues. And they can release inflammatory mediators, but they are not as potent with respect to releasing inflammatory mediators that I'm going to tell you about in the next slide when we talk about gram-negative bacteria. But I just want to remind you, gram-positive is a thick peptidoglycan layer, multiple cross-links through all the layers, and side by side, all the strands are cross-linked into that rigid cell wall that makes up a gram-positive bacteria. Now, when we look at a gram-negative bacteria, which isn't stained by gentian violet, okay, that stain washes out, what we see in that is a very, very thin, only 1 or 2 layer thin of peptidoglycan shown here. It is cross-linked, okay, but it's very thin. And as a result, when you do a gram stain, whatever gentian violet gets in kind of washes out. And then we use the safranin stain to stain, actually binds to lipids and binds, and it stains all bacteria, but it's pink, and the blue predominates for the gram-positive bacteria. So gram-positive bacteria essentially overwhelm the pink stain, but the gram-negative, which has lost and had that gram stain washed out of it, will now be stained pink. Hence, blue, dark blue for gram-positive, pink for gram-negative. So we have this thin peptidoglycan layer, but on the outside of that layer is a second membrane called the LPS, or lipo, and it's made out of a lipid bilayer shown here, as well as polysaccharide shown here. Now, it turns out that this LPS is also known as endotoxin because it is part of the bacteria. And this is one of the most toxic parts of a gram-negative bacteria. It's why gram-negative bacteria are so much more dangerous in many respects than gram-positive bacteria, because this endotoxin can elicit a sepsis reaction. Okay, now remember, I told you gram-negative, gram-positives can also do it, but not as efficiently as this endotoxin. Okay, in addition, we have proteins that, like porins, that enable stuff to get through into the membrane, into the periplasm, the space between the LPS and the actual plasma membrane. We have these porins. And when we are talking about lipopolysaccharide, what we see here is that outer lipopolysaccharide. So we have a lipid bilayer over here. And attached to that are sugar phosphate molecules, which we call the inner core sugar phosphate. And then we have the outer core polysaccharide. Okay, this is referred to as the O antigen, and this is referred to as lipid A. And lipid A is the highly toxic form, the real toxic part of that endotoxin. And the reason it's toxic is that we have a systemic inflammatory response to LPS, to endotoxin. And that's because basically when bacteria are growing, they're always growing, they're getting old, they're lysing. And even if they don't lyse, some of that LPS gets sloughed off and is found in the bloodstream or in infected parts of pus and tissue, et cetera. And we have an LPS binding protein shown here, okay, that will bind to it. And this LPS binding protein's job is to essentially to stimulate macrophages by lipid LPS, LPS binding protein, LPS complexes will bind to CD14, this toll-like receptor, okay, and will induce a cascade of cytokines. Now, you've all heard this cytokine cascade when we're talking about COVID, okay? Now, cytokines are really important because they bring on the passive immune response that is going to help get rid of bacteria, except if it goes a little bit rampant and you start making too much of that passive immune response. This is an immunology course, so I'm not going to tell you much about it, but that passive immune response that usually brings on inflammatory responses that help get rid of the bacteria can bring on a massive inflammatory response, which we refer to as sepsis or poisoning. And as I mentioned, gram negatives have a very toxic LPS, whereas teichoic acids, as well as some fungi, produce a much less severe response, not quite the kind of sepsis. And when we're talking about sepsis,
Dr. Kaback, sorry, before you move on,
okay,
there's a question. Could you repeat which region is considered the O antigen?
Okay. The O antigen, okay, is the outer part. Okay, so showing here. This is the O antigen, and this is lipid A. Okay, so the O antigen, which is what's seen by the antibodies to it, is really recognized mostly via this whatever kind of polysaccharide you have over here. Okay.
Thank you.
All right. So sepsis, basically, if you suspect bacteremia, maybe person comes in with a high fever, spiking fever, okay, or they're cold, maybe they have a rapid heartbeat, rapid breathing, hypotension, and then a multitude of other than symptoms you suspect sepsis, okay, basically low systemic vascular dilated arterial system, so blood pressure is very low, high cardiac output, the heart is beating very fast, all sorts of things for diagnosing sepsis, and seeing a bunch of these syndromes, you suspect a bacterial sepsis, and you want to treat that patient very, very aggressively. And so here's a patient, they had fever, tachycardia, arterial hypoxia, their oxygen level, because they were having trouble breathing. They were actually undergoing acute renal failure. They were basically passing out, and so they had all the signs of sepsis. So you need to rapidly treat when you see somebody with that. All right.
David, just on the issue of sepsis as a second question, can you clarify, do small amounts of LPS cause sepsis, or is an accumulation required?
Again, that is a very, very tough question to answer. Okay, I don't know of the quantitation. It's not a lot. Okay, and it depends where and when and what's seeing it. Okay, so
Sounds good.
so small amounts doesn't mean anything. That's a good question to ask an immunologist, is how much LPS is required. You can look that up. Okay, but I don't know exactly when, but it has to do where and when the infection is, the immune state of the patient. It's probably there's probably no good answer to it. Okay, all right, let's bring on the next question, the next slide. Just a little review of what you need to know about the structure of gram-negative and gram-positive organisms, which brings me to the next thing about cell wall, is that many bacteria have many different kinds of cell walls based on the structure, whereas they all have this general structure of glycan, cross-linked glycan with diamino acids. Gram-negative bacteria usually have the two cross-linking peptide directly linked to each other, cross-linked to each other via a peptide bond between two amino acids. Okay, whereas gram-positive bacteria actually often have an adapter peptide. Okay, so here is staph, and staph actually, the peptido portion shown here on two different chains. We have a chain here and a chain here, and the peptido portion shown here and the peptido portion shown here. These are actually cross-linked together via pentaglycine bridge. Okay, 5 glycine residues come in and are cross-link these. And these are all enzymatically made. They're not made by protein synthesis. They're made via an enzymatic reaction to produce them. All right, so there's tremendous diversity. Even a single organism like Staph aureus has different alternative positions of these peptido portion depending on the strain, and you can see these alternate positions. Again, this is not a starred slide, so it's nothing you need to remember other than that there is diversity in the way these peptidoglycans are cross-linked together in both the sequence and form that they are. Okay, now the cross-linking and the structure is very important for antibiotic sensitivity. So for example, penicillin, okay, penicillin, which is, by the way, very effective for gram-positive organisms, usually, something you can kind of remember, and vancomycin is involved in, it prevents the cross-linking of adjacent polypeptides on adjacent strands. Vancomycin also prevents cross-linking, okay? In addition, vancomycin actually affects the polymerization of the polymer, okay? So it has multiple targets, multiple enzymatic targets that it affects, okay? Bacitracin is involved in recycling the synthetic apparatus, again, something you don't have to really remember.
Okay, everybody can hear me too, right? So Dr. Kaback is frozen for me. I'm just going to text him now and let him know. So everybody just bear with me a little bit while usually Dr. Kaback realizes if he freezes and he'll log out, come back on.
It's modern problems of your internet blowing out, and we can blame Optimum. Okay, now in the old days, it was great because not only could you blame Optimum for messing up and dropping your internet connection, but the same people who owned Optimum also owned the NICs. And boy, if you want to cast blame on a family for both dropping your internet connection and the NICs, it was very easy. Unfortunately, can't do that anymore because he sold Optimum. So I think he's only at fault for dealing for the NICs at this point, and you can only blame him for the NICs. At this point, Optimum is owned by a Dutch company that has nothing to do with the same family that owns the NICs. Okay, that said,
David, can you
Yeah.
start, at this point, I think what you've recorded so far is being processed, which is fine. I just gave you permission to start recording again. So can you record the rest of your material? You should be able to start
it looks
recording.
like I am recording. I'll double check.
No, you're not.
No. Okay. Yes. Okay. Record.
Now you are. Okay, good. Thank you.
Okay. Wonderful. All right. So the last thing I spoke about was that the cell wall structures very much affects antibiotics sensitivity because penicillin, for example, is involved in inhibiting the transpeptidation reaction. So a cell growing in the presence of penicillin cannot do this cross-linking. The cell wall becomes very weak and non-effective and virtually non-existent. Vancomycin inhibits both transpeptidation, this cross-linking reaction, but also the polymerization of that polymer, that glucose polymer shown here. Bacitracin is involved in the recycling, affects the recycling of proteins that are involved in producing this reaction, this polymer. So this kind of explains one of the things it explains is why gram positives are so sensitive to penicillin, is that they have very thick cell wall that needs to be there because they don't have an outer membrane, for example. All right, that said, if you grow a number of organisms in the presence of penicillin, it will actually produce a cell that lacks a cell wall or has a very flimsy cell wall. And that cell becomes osmotically sensitive so that if you put that cell in hypotonic media, that cell will lyse, okay? And on the other hand, if you stabilize the cell and put it in isotonic media so it won't swell up and lyse, that cell will actually be capable of growing, and that's called a spheroplast or a protoplast, okay? Not terribly important. At one point, it was thought that this might be a mechanism of drug resistance, that cells could grow in absence of a cell wall, but apparently now it's not thought to be important clinically, that basically you really can find what are called L forms in infection of cells growing without a cell wall. And I show this because without a cell wall, basically a cell will lyse, okay? But you can get a limited amount of growth if you stabilize the osmotic media with making a high osmotic pressure so the cell doesn't explode like a balloon. Okay, finally, one last cell envelope review to show you how important it is to know the different structure of gram negative and gram positive cells. And it is, let's continue on for 2 more minutes and then we'll have another break. Okay. All right. In addition to cell walls, we have auxiliary extracellular or cell surface components. Okay, we talked about the cell wall, which is required for osmotic stability, but we also have excess polysaccharide, which we refer to as the glycocalyx. And this is, glycocalyx refers to the fact that bacteria very often have these very thick layers of polysaccharide, which we refer to as capsules. And very often they produce so much of this polysaccharide that they form biofilms, literally things that you can scrape off. And a best way to think about biofilms is slime, okay? And they have pathological circumstances I'll show you. And the glycocalyx, whether they're capsules or biofilms, are important for preventing phagocytosis, the macrophage eating the bacteria. They inhibit immune surveillance by preventing T cells from seeing the actual surface of that bacteria and making T cell receptors or antibodies against that bacteria. And furthermore, they form a barrier that prevents the penetration of some antibiotics into the bacterial cell where it can be taken up and kill that bacterial cell. Okay, so this is the glycocalyx, as well as flagellae and pili. But let's talk about the glycocalyx. Okay, here is a capsule. And when a cell is making a capsule, as shown here, okay, and here you can see the lipoteichoic acids, for example, on the outside. And sometimes we refer to the capsule as this capsular polysaccharide shown here, sorry. When a cell is growing colonially, and it's making a capsule, very often you will see this jelly-like thing, and these are very often called mucoid colonies because they look like mucus. The colony growing will be what's referred to. And these generally, if you were to essentially put this in make a slide from that, the cells would still be distinct, okay? And they would be able to fly off one another because the capsule has a well-defined border, okay? And capsules, as I mentioned, are very often antiphagocytic, as I'll show you in a minute. So that's the capsule. If it's making a biofilm, it's also made out of a polysaccharide polymer, secretion of large amounts of a polysaccharide polymer. And in this case, the cells are much more like a biofilm. They're much less soluble. You don't see them, if you were to shake a biofilm, it wouldn't disperse into single cells. Certainly single cells would come off, but it wouldn't sort of become easy to homogenize. Whereas something that's encapsulated, if you shake it up, would be much easier to disperse those cells. So when you look at a slime or a biofilm layer under the microscope, you would see something like this, where the polysaccharide form a mesh-like behavior, have a mesh-like behavior where the bacteria are embedded in that. And that very often can prevent antibiotics from penetrating it. And one of the classic biofilms that we see pathologically, am I off again? Hello?
Yeah, you're fine, David. I don't know what that is. I heard that too, but you're fine.
Okay. Yeah, I don't know what caused that. Okay. So in Corynebacterium diphtheria, which is a respiratory pathogen, you will form this biofilm on the throat. It's called a pseudomembrane that you can literally pick off with a set of tweezers from somebody who has diphtheria. So you can see that biofilm there, Okay, so when we have a bacterial capsule, okay, what happens now, normally in the absence of a capsule, okay, antibodies or a complement protein called C3b will coat that bacteria. This is a passive immune response. This is an active immune response where you've made an antibody to that bacteria. That either C3b or that antibody will coat that bacteria. This coat is referred to as opsonization. Okay, when a bacteria is opsonized by either C3b or an antibody to its cell surface, a macrophage will now recognize these proteins that are on the surface of it and engulf that bacteria and swallow it up. And the lethal, after phagocytosis, you get cell lysis, will happen, and the bacteria will be killed by the macrophage. Okay, so you don't, for a bacterial infection, the bacteria needs to avoid being swallowed up by a macrophage before it causes any pathological symptoms. On the other hand, if a bacteria has a capsule, okay, if a bacteria has a capsule, it can easily be opsonized, as we've seen here by these opsonizing proteins, C3b or an antibody. Okay. On the other hand, if it has a capsule, the antibodies with C3b will get through that capsule. On the other hand, it has this thick polysaccharide jelly on the outside. So whereas this guy that's coated and opsonized by these proteins can be easily recognized by a phagocyte, shown here, and bind to it, so it then gets swallowed up by it. The encapsulated bacteria, shown here, is much harder to phagocytose because the macrophage cannot see those opsonizing proteins and recognize them and induce that phagocytosis. So the capsule is a very important part of preventing phagocytosis for those encapsulated organisms. And you're going to hear about many, many pathogens, and you'll hear for those many, many pathogens, it's an encapsulated gram-positive organism, an encapsulated gram-negative organism. So capsules are very important virulence factors. In addition, on the outside of cells, bacteria have flagella. Okay, and these flagella are important for motility because they enable that bacteria to swim out in the environment and essentially find other cells. And these actually are characterized as being antigenic, and you'll see a number of bacteria, when they talk about an H antigen, they're talking about the protein flagellin, shown here, which is actually a fairly well-conserved protein that is the major protein of this motile structure that can cause the bacteria to have motility. Okay, and finally, we're talking about pili. Okay, these are also proteinaceous filamentous appendages shown graphically here, and here, that are involved in a number of different functions, and it depends on the bacteria and the protein, but they are very often involved in cell adhesion to a when the bacteria is pathogenic and needs to adhere to a mammalian or human surface. So they can very often become virulence factors. There are also a number of pili that are involved in bacterial sex, that are involved in transferring DNA, that you may become familiar with in certain bacteria. And there are other pili that are involved in actually taking up DNA directly from the outside. So they're very common for many bacteria to have pili. And in fact, I just learned last week that normal flora, a number of normal flora, there are Neisseria species that are part of our normal flora. And one of the ways that they prevent pathogenesis by Neisseria gonorrhea is that these strains of these species of Neisseria actually will inject their own DNA into Neisseria gonorrhea and cause that bacteria to destroy the Neisseria gonorrhea to actually destroy their own genomes. Rather unique way that our normal flora actually can get rid of some abnormal flora. This just came out. All right. That said, pili are important for adhesion. This experiment kind of shows you this, where we took Neisseria and we put it in a blender, and that made the pili actually come off by that hydrodynamic shear, got rid of the pili. And when you look at the bacteria attached to a regular cell that hadn't been put through the blender, you can see bacteria easily attached to the epithelial cell, shown here, these dark spots of the bacteria, whereas the depilated, I guess it would be, remember Nair, the depilated bacteria do not adhere to the epithelial cell. Okay, that said, I think we'll take another 5-minute break, if you don't mind. And then we'll come back for another 20 minutes or so.
All right, so it's 4:08. Why don't we'll come back on at 4:13 if everybody wants to get up, stretch their legs a bit, and we'll start off in 5.
I just paused the recording.
Good.
Zoom recording. Hi, everybody. Hopefully you're all back. I just started the recording.
Basma, go ahead and turn on your mic if you have a question.
Hello, Professor. Can you hear me?
Sure.
Okay, so I have a question on an earlier topic. It's regarding penicillin's mechanism of action.
Yes.
I understand that penicillin will act on the enzyme responsible for transpeptidation. However, if you were to follow the life of a bacterial cell following the administration of penicillin,
Yes.
what exactly would cause the ultimate death of the bacterial cell? Because I can imagine mitosis might be difficult if you can't create more cell wall, but I don't exactly
Good point.
understand a protoplast, like
Okay.
how
So
you get there.
basically, if a cell sees itself in the presence of penicillin, number one, it has to be growing. Okay, penicillin will not kill a cell that's not growing. Okay, so if there's no media around, you can put all the penicillin in the world there, it wouldn't do anything. Okay, if it's growing, it would enlarge. It would enlarge without a functional cell wall, and basically and most media is hypotonic. Even blood, the concentration of salt and ions and small molecules inside a cell is much higher than outside a cell, even blood cells, okay? It's not isotonic. So, and as a result, virtually any bacteria would swell in virtually any kind of tissue. Does that answer your question? And would ultimately lyse.
I think my question has more to do with if there's already a bacterial cell wall, like the integrity is there, and we have a full 40 layers of peptidoglycan, will penicillin do anything to this? Do you just have to wait for the natural death of these bacterial cells?
No. In other words, remember that a bacterial cell, when it's put in media, is first of all, mitosis is something only eukaryotic organisms undergo because mitosis requires a mitotic spindle. But cell division, okay? Anytime a bacteria is undergoing cell division, it's going to undergo enlargement and then rupture. Does it take 2 generations to kill the bacteria? Probably. Okay. In other words, it wouldn't lice immediately, but it would start, there would be weak, virtually wherever so there would be weak parts of that bacteria, especially at the place where the septum is forming, okay, where new cell wall material is required that wouldn't have a cell wall. As soon as you inhibited transpeptidation, you would inhibit that part in that part of the cell, and there water would get in the cell and the membrane would just swell around it. It would be kind of like having a balloon and it blebbing out. I mean, I'm sure if you take a balloon and you hold your finger and squeeze it, part of it will pop through the finger, like when you're making a balloon animal. You understand that?
Yeah, that clarified. Thank you very much.
Okay.
So David, you're saying it wouldn't kill an existing cell, but it would prevent the cell from growing and dividing. Is that correct?
Well, no, it would kill that cell. It would not kill a cell that is not actively dividing, but bacteria are always if a bacteria,
They're always actively dividing.
medium,
So, yeah, okay.
if a bacteria has media and that it has adapted to, okay, and that's an important part of the next lecture that we'll hear about in the next hour, is that there is a stage of bacterial growth where the bacteria is dormant, where it's basically adapting to its kind of media. And at that point in the bacteria's life cycle, it will actually be resistant to penicillin until it starts growing again. Okay, so you'll hear that in the next hour. It's a great question. All right, so I am recording, I hope. Yes, Ian, I'm recording at this point.
Yes, I see you recording. Yeah.
Great. All right, so that said, let us go back to our structure of the bacteria. And now what we're going to talk about is a little bit about growth of bacteria and going back to that bacterial genome in the next slide. Okay. Just want to show you that and remind you that bacteria grow by binary fission, not by mitosis, as I mentioned in the question. Mitosis refers specifically to a mitotic apparatus made out of microtubules that separates mitotic chromosomes. Bacteria grow by binary fission, and they separate, which is not using a spindle. They pull apart their chromosomes due to their attachment to the membrane as it's expanding out this way. Okay, and bacteria grow exponentially. Now, different bacteria have different growth times, okay? So for example, E. coli and Pseudomonas, these are rather rapid growing bacteria that can double in 20 minutes. In contrast, Mycobacterium tuberculosis, even under the best condition, takes about 24 hours to undergo a cell division. Mycobacterium leprae that causes leprosy takes 7 days to undergo a doubling. So there's tremendous heterogeneity in what we refer to as generation time, which affects the rate of growth of a bacterial culture or the rate of growth of bacteria in a person. Irrespective of that, bacteria grow exponentially, which can be described by a growth curve of that organism or of that culture. And when we grow bacteria in culture, we discover that when you put it first in whatever media, you take it out of one growth media and put it in another kind of growth media. And essentially it says, I'm in a new home. I have to adapt to growing under those conditions. So for example, if it's in the bloodstream, it may not have the amount of glucose necessary for it to grow rapidly on this, and it may require new enzymes. If it goes from, if it comes from underneath your arms where it's growing mostly anaerobically and it has to adapt to aerobic conditions when you're putting it in culture, it may have to synthesize new enzymes in order to grow, to adapt to that new condition. And during that time, a bacterial culture will undergo a lag phase where you don't see cell division. This is referred to as lag phase. But once it is adapted, each cell will undergo a doubling and it will grow exponentially. And we refer to that as the log phase or exponential phase of growth, where cells essentially grow much more rapidly than the non-growing lag adaptation phase. When they run out of conditions, usually food, so that they can't grow anymore, and sometimes it's because they actually produce peptides that tell the cell that there's enough in the culture to stop growing. They actually signal to each other that, hey, we're running out of food, let's stop growing. They enter what we refer to as stationary phase. And not all bacteria have this signaling mechanism, but some do. And that's called stationary phase, where we don't see apparent growth. Actually, very often the cells start dying. And sometimes we see that that the cell death is equal to the new growth phase, and the cell is in stationary phase and the number of bacteria remain constant. Okay, now, as I just mentioned before, antibiotics are not likely to react. Ones that affect cell walls are not likely to kill a cell in lag phase, but will certainly kill cells and cultures when they are in exponential phase. Whereas a protein synthesis inhibitor, which is going to affect the ability to adapt, of that bacteria to adapt, may be effective in killing a bacteria or preventing that bacteria's growth while it is adapting and can't make that protein that's going to enable it to grow in the new, under the new conditions. So the phase that a bacteria is in, whether non-actively growing or actively growing or dying at a certain phase, is going to affect the resistance or sensitivity to certain antibiotics. Okay, so this is an important slide. Lag phase, log, stationary, and death phase. Understand what they are. Okay, then bacteria have very different nutritional requirements depending on what kind of bacteria it is. Most bacteria are what we refer to as heterotrophic. They require some kind of carbon source, reduced carbon source like glucose, but they may require something like, they may be able to grow using ethanol as a carbon source or lactic acid as a carbon source. If they do that, they're going to require oxidative phosphorylation and a cytochrome system in order to do that. Okay, these are referred to as heterotrophic. Okay. A few bacteria are autotrophic. They undergo photosynthesis. These are the blue-green algae. They're able to make their own, they're able to reduce their own carbon system for, in order to grow on. These are the blue-green algae. Fortunately, I don't know of any pathogenic blue-green algae that are autotrophic. So virtually anything you hear about is going to be heterotrophic. Now, some bacteria can actually grow on what we refer to as minimal media. Okay, this is just glucose, ammonia for nitrogen for making proteins and nucleic acid, sulfate, phosphate, as well as a number of trace metals that are cofactors for enzymes. Okay, some bacteria can grow on this minimal media, okay, and can grow on agar containing what I just told you. Most of our pathogens require a little bit more complicated media. And very often undefined media where we use protein hydrolysates, yeast extract, things like that. Trypticase soy agar is the reference agar for growing bacteria that has blood in it. So we don't really know what's in it, but it's a complex mixture of things that some bacteria require. And there are some bacteria that require things like NAD and hemin in order to grow. Okay, so there are a whole lot of different requirements. Now, some bacteria cannot at all be cultured in cell-free media. They may require intracellular growth, so we grow them in cell culture, mammalian cell culture. And things like Mycobacterium leprae, we can't even, the only place we can grow it, this causes leprosy, the only place we can really grow it now is in armadillos, believe it or not. Okay, we have to infect an armadillo and harvest the bacteria from armadillo. Treponema pallidum, which causes syphilis, we can still only grow in mammalian hosts, usually rodents, rabbits. Okay, and there are some bacteria, no matter what we do, these are obligate intracellular bacteria, and they can only be cultured in mammalian cells, okay? Treponema and Mycobacteria, these are not obligate intracellular organisms. They grow within the organisms, but not inside the organism cells. And I'll tell you about chlamydia and rickettsia that can only be grown within a cell of an organism, okay? So highly variable types of cell growth. Okay, and that's due to the fact that some bacteria have the enzymes to synthesize all the amino acids, all 20 amino acids. They can essentially take sugar and turn that with a little ammonia into all those amino acids via biochemical pathways. Whereas other bacteria basically as I mentioned, require things like hemin. In general, a lot of bacteria are synthesizing proteases and hydrolases that are able to dissolve the macromolecules around them, which is an important part of the pathogenic process, okay? So if they see tissue and they invade it, they actually feed upon it by dissolving it and turning it into small molecules, amino acids and sugar, and taking those up and living on that. Again, highly polymorphic. Things like, and we use these differences to diagnose. So for example, one of the ways we differentiate between Neisseria meningitidis and Neisseria gonorrhea is Neisseria meningitidis grows on the disaccharide maltose as well as glucose, whereas Neisseria gonorrhea can only grow in the presence if you give it glucose. You give it maltose as a carbon source, and maltose is just a disaccharide of glucose. Okay, you give it maltose and it won't grow. Whereas Neisseria meningitidis, if you give it just maltose, it grows just fine. So that enables us to tell the difference between a gram-negative rod, Neisseria is gram-negative rod, if I recall. I'll remember that in a minute. That is oxidase positive, that makes cytochrome oxidase. And both these two are, but only Neisseria gonorrhea will not grow on maltose. So that enables us to differentiate them. So these characteristics. Now, is this important for the future? No. Is it important for the present? Yes. People
Okay.
still.
I found this on the web for
Oh my God. I didn't say Siri. God. Siri interrupts me. Terrible. Anyway, that said, in the old days, this is the only way we would differentiate bacteria. We had all these biochemical tests that we put out on these grids like this, and we could read off depending on whether an organism ferments a certain kind or doesn't ferment a certain kind. And we would get these readouts on these what are called API strips that would enable us to differentiate between different kinds of bacteria based on metabolism. Now we can do PCR and it's much faster. This takes 2 days to do the full analysis, 1 day to get a rapid number, 2 days to do the full analysis. PCR takes 45 minutes to see if it is. So again, this is for analysis going by the wayside as we are advancing into the genomic age. But nevertheless, the important thing to remember about that is different bacteria have different metabolisms that, of course, provide a good reason for them establishing themselves in certain tissues or certain parts of your body and being pathogenic if they get into the wrong parts of the body versus others that are totally benign in certain parts of the body. Okay, and these conditions are not only nutrition, but they're also pH of certain areas. We have acidophiles like lactobacillus that grow in an acidic environment. Vibrio requires it. Certain strains of Vibrio require an alkaline environment. Most bacteria, like people, really prefer a neutral pH, by the way. We have anaerobes and aerobes, and of course, facultative anaerobes, ones that can grow aerobically because they can make cytochromes and do oxidative phosphorylation, but they can also grow using glycolysis or another alternate pathway that can make ATP. Temperature requirements. These organisms require different temperatures, and things like E. coli can grow at 20 degrees, but can also grow at 37 degrees. And as a result, we find them growing in swimming pools, okay? In contrast, things like listeria. Listeria can grow at 4 degrees. It can grow in the refrigerator, which is why we have to wash our lettuce because you don't want listeria growing on your lettuce. It's a normal ground organism that we find, and you want to wash your lettuce to make sure, especially if you leave it in the fridge for a week. So we'll hear much more about that later. Osmolarity, we have organisms that like salt and others that require an absolutely solid osmotic pressure. So bacterial diversity is a very important aspect of where, when, and how often different organisms grow, when and where, and in different parts of the biosphere, and an important part of bacteriology. And as I mentioned, mycobacteria has a slow bacteria growing time. This causes TB. Pseudomonas, which causes lung infections, can grow in less than 20 minutes. It has a 20-minute doubling time. Same thing with E. coli in the right environment. Okay, so this is what I just told you. Different organisms are found in different places, both in the wild and in humans. And positive identification of an organism, which is based on all of the characteristics I told you, enables you to affect the outcome of dealing with an infection. It enables you to treat with the right antibiotic at the right time. Finally, I also want to tell you about endospore formation. Is that certain bacteria, these are known as spore-forming organisms. Okay, a lot of them are anaerobes, but some are aerobes. Essentially, when they run out of food or see a harsh environment, instead of undergoing a normal cell doubling like we see here, undergo a differentiation where they take one of their genomes after DNA replication and send it over to a specific part of the cell where they essentially form a new cell inside the old cell. Instead of having a cell divide, it forms a new cell inside the old cell with a plasma membrane and then a cell wall around that. We're still inside the old cell wall. And then we pump all the water out of that cell, virtually all the water, and we have a substance called dipicolinic acid, or calcium dipicolinate is the salt, that essentially enables that cell to protect all the macromolecules and the nucleic acid within that cell. That cell has virtually no water in it, and it becomes very resistant to drying and essentially is air stable and can survive for hundreds of years. So for example, Louis Pasteur, who worked on Bacillus anthracis, a spore-forming organism that causes anthrax, the spores from his lab at the Pasteur Institute, it wasn't the Pasteur when he worked there, but his lab in Paris, okay, those spores every so often they will check to see, and those spores 170 years after working on them, those spores are still viable. If you take them and plate them out, they will still form colonies of the anthrax bacillus, okay? So they're highly resistant to drying. They can even be autoclaved and not killed in an autoclave, high boiling temperature, dry steam treatment, and they still are somewhat viable to autoclaving. The only way to truly get rid of anthrax spores is to incinerate them. Okay, and we'll be hearing more about that later on. Okay, so spore formation is an important mechanism how bacteria survive under extremely harsh conditions. Okay, now I just want to...
David, there's a question. Are
Okay.
there any antibiotics which target the endospore itself, or do endospores not typically exist in the body?
The answer is, I do not know the answer to that, whether any I don't see how they could. They may inhibit, certainly a protein synthesis inhibitor will prevent a spore from forming, okay? And will probably prevent a spore from germinating. Almost certainly prevent a spore from forming another spore once it is. But to inhibit the process of sporulation directly and not inhibit other factors, probably, I don't know of any. I hope that answers the question. Okay, but a protein synthesis inhibitor will certainly prevent sporulation. Okay, hope that answers your question. All right, let's move on. Okay, just want to tell you a little bit more about bacterial genomes very quickly, and then we will be finished. Okay, here is the bacterial chromosome. I want to remind you that we have the bacterial chromosome and we have plasmids, shown here, extrabacterial elements that grow in parallel to these chromosomes. These very often are involved, and I mentioned this before, in sex or in drug resistance mechanisms. They very often carry the genes that encode the enzymes that enable a cell to detoxify penicillin or to prevent certain other antibiotics from doing their thing. And because they are extra chromosomal DNA molecules, they can move between bacterial of the same species as well as different bacterial species. So they're very important. In addition, bacterial elements have what are called transposons on them that are involved, able to replicate and move themselves to different parts of the genome or onto plasmids. And these transposons form another mechanism by which bacteria are able to easily exchange DNA between them. Okay, now when a bacteria, okay, oh, by the way, Borrelia, interestingly enough, has a linear DNA molecule as opposed to a circular DNA molecule. Just sort of an interesting curiosity. And its plasmids are also linear. And that's kind of an interesting anomaly that we find. And Vibrio cholera actually has 2 circular chromosomes in order to provide life. So nothing is set in stone that a bacteria has only 1 chromosome and it has to be a circular DNA molecule. Most bacteria have a single chromosome consisting of a circular DNA molecule. When that chromosome replicates, it usually does so by starting at a single origin of DNA replication, and that origin and DNA replication proceeds bidirectionally, meaning that at that origin we replicate going this way and going that way. And just to review from fundamentals, when a bacterial chromosome replicates, it goes in one direction on one strand, synthesizing 5' to 3' in this direction, and 5' to 3' in that direction, because DNA is a double helix that has the two strands in opposite orientation, and we have a leading and a lagging strand. Okay, this is just a little biochemistry. Okay, and basically, genetic variability is an important part of bacterial pathogenesis, because bacteria evolve because they replicate so much. They actually evolve very quickly. They can transfer genes, especially antibiotic resistance genes, between organisms, which is why bacterial antibiotic resistance is such a worrisome factor. And bacteria can become resistant to antibiotics both by mutagenesis. So if a ribosomal protein mutates, it may make that ribosome immune and not subject to inhibition by something like streptomycin, which is an antibiotic that inhibits the ribosome. Or it may transfer one of these resistance factors to another bacteria. And the reason that bacteria have these genetic resistance factor, these did not come about because man discovered antibiotics. Okay, antibiotics have been around since fungi and different strains of bacteria like streptomyces or actinomyces have been living in the environment in order to provide their own bacterial niches where they can grow in the environment. They produce toxins that kill all the bacteria or fungi that are surrounding them, so that they are resistant to their own antibiotics, but they kill all the bacteria around them. And we discovered that in the 30s with penicillin, that penicillin mold, in order for it to grow without being taken over by bacteria, that ecological niche being taken over by a bacteria, which was much faster growing than it, they made penicillin that would kill all the bacteria around it, so that they could basically eat the proteins, et cetera, that are in the wild and have their own bacterial niche. Okay, so horizontal gene so bacteria, of course, fought back by producing drug-resistant genes and proteins. They evolved proteins that enabled them to detoxify various antibiotics. Okay, and antibiotics, as I say, are made. Penicillin is made by a fungus, but streptomycin is made by another bacteria. So essentially, we have this ecological interplay, and different species can transform different things, and bacteria, in order to transfer genes, have evolved mechanisms, and we have sex in bacteria where they can actually form physical constructs and transfer their DNA from one bacteria to another. Some bacteria, as I mentioned before, will uptake and integrate DNA into their systems. This is the mechanism how I'm sure most of you have heard of CRISPR-Cas9 leading to resistance of bacteria to certain viruses that we've now used for our sake for genetic engineering. Transduction, bacteriophages, viruses can move parts of bacterial genomes from one bacteria to another. Okay, now when we're probably the most important thing to talk about here is the fact that bacterial plasmids carry drug resistance markers on them. And here you see streptomycin, chloramphenicol. Bacterial plasmids very often contain multiple genes that each detoxify a number of different antibiotics, shown here, kanamycin, chloramphenicol, as well as streptomycin on these plasmids. And these referred to as R plasmids for resistance plasmids, and they produce simultaneous resistance to several antibiotics. Okay, and we have conjugative plasmids that are involved in mechanisms for specifically moving genes between usually the same species of a bacteria. Okay, and then we also have virulence plasmids that enable certain strains of bacteria to become virulent. And I'll tell you about the plague bacteria containing a number of virulence plasmids. Okay, and when we talk about conjugation, we use pili to transfer DNA, to recognize and bind and then transfer that DNA from one cell to the other. This is called conjugation. It's very efficient. Okay, and when we transfer the genes for a male cell into a female bacteria that doesn't have those genes, that bacteria now becomes a male cell. Okay, so conjugation is an important part of the sexual cycle of bacteria, and a very important aspect of this process is breakage and reunion of DNA, and that's called recombination, where we have homologous recombination and non-homologous integration. Homologous recombination requires homology between the two DNA sequences, whereas non-homologous recombination just requires that piece of DNA to jump into a new spot in the bacterial genome. And it's the process of transformation that was initially found in pneumococcus that enabled us to show that DNA was the genetic material. Okay, and I'm just going to run through these slides very quickly because it's time to go. Okay, so you can look at it. But these experiments where we essentially took a strain of bacteria that was non-pathogenic because it didn't have a capsule and was able to introduce the DNA for producing capsules into that cell, and that cell became pathogenic, able to cause this pneumococcus to kill the mouse over here. Okay, and that process is called transformation, where DNA is taken up by the bacteria, shown here, and integrates via recombination. So here's that DNA coming into the cell and undergoing a crossover event to integrate that sequence into the genome, shown here. Finally, non-homologous integration, which doesn't require DNA homology, that's done by transposons, is that this piece of DNA can essentially replicate and move itself into a new part of the same chromosome or into a plasmid. Okay, this is called non-homologous or illegitimate recombination. And the things that do that best are called transposons that very often carry drug resistance markers. Okay, and that's pretty much all I have to tell you about. And now, whereas bacteria are dependent upon media in order to grow, our own brains are also dependent upon intellectual media. And I just want to give you a commercial for if you're totally into microbes, as I am, I want to recommend some human brain growth media. One is the book Arrowsmith, okay? Anybody who has any desire to go into the health professions should read this novel by Sinclair Lewis, okay? It talks about dedication to, in this case, research and medicine, as the main character, Martin Arrowsmith, had. It's a great novel. It was actually half written by Paul de Kruif. Now, Paul de Kruif wrote a book called Microbe Hunters about the initial people who were discovering all these new microbes in the turn of the century that caused disease. And actually, Sinclair Lewis, who was a famous author of Babbitt and Main Street, if you know, essentially because of Paul de Kruif's contribution to Arrowsmith and the fact that his books didn't sell that well, he actually gave Sinclair Lewis a quarter of the royalties of Arrowsmith. Sinclair Lewis gave Paul de Kruif some of the royalties. Now, if you don't like reading and you want to have a fun movie, go on Turner Classic Movies and watch Dr. Ehrlich's Magic Bullet about the predecessor of the acid fast stain, and a great role of Edward G. Robinson. Now you're probably all familiar with Edward G. Robinson with playing Little Caesar in the movie Little Caesar, where he plays a gangster, or in the movie Key Largo with Humphrey Bogart. Here he plays a nice guy, an educated doctor. It's great. It was made in 1940. It's got all this anti-Nazi stuff in it. It's a terrific old movie. It's available on Turner Classic Movies, as is the story of Louis Pasteur, where he talks a lot about anthrax and making the first vaccines for rabies. Another great movie. And in fact, Paul Muni won the Oscar for Best Actor for this movie. All right, as well as 2 more Oscars, and it was nominated for the Best Picture. So these are great movies if you want to learn about microbiology. And with that, I'll see you. If there are any questions, I'll take those. Otherwise, I'll see you in a couple of weeks when we talk about pneumonia. All right.
Okay, so why don't we go ahead and take any questions? We'll keep the recordings going, David, until we're finished the question period. Feel free to, if you have a question for Dr. Kaback, feel free to unmute. And
All right, how many people? We still have 40 people on. I don't see anybody. 39. Do a countdown.
all right, that's fine. If there's no questions at this point, that's terrific. And as I say, just
Email me
yep.
your questions.
Now, with regard to
If I can't answer them in a brief email, I may ask you to, what you might do if you're emailing me a question is include, if I can't email a quick answer, include your cell phone number, and if it's an iPhone, and whether you do FaceTime. Otherwise, I will invite you to a Zoom conversation and answer it in however long it takes via Zoom. But if you have an iPhone and can do FaceTime, send me your cell phone number and I'll just I do FaceTime with you if I can't answer an email quickly. So include your cell phone number in any emails. All right, I'm stopping the recording. Let's ask everybody to get off at this point. And Ian, stay on if you don't mind.
sure.
Okay, I just stopped my recording, I hope, right?
Have a good night. Thank
All
you.
right, good night, everybody. Ian, stay on.
Yep.